Hi, everyone. Welcome to the first week of Intro to Programming in Bioinformatics. In this video, we're going to look at some really common file types, and we're just going to look at the data structures that are most commonly used to store those bioinformatics file types in R. One of the things I want to introduce you to is the APE package. It's got a lot of great built-in tools that will be useful for us throughout the semester. And I also just want you to start thinking about one of the good practices for each of your scripts to use the str struct command to figure out how the data is being stored and question whether or not you're choosing the data structure that's going to give you the best way to store the data in terms of memory usage, but also in terms of being able to run inquiries on the data. And one of the things I think you'll see with the way that the functions have been built into APE is that you get a lot of really useful information from the sequence that you would want as a biologist and as a bioinformatician. So the first thing we're going to do is go to Tools, Install Packages. You're going to want to make sure that you have APE installed. And then you can check that. I usually try to put the library commands in my R setup of my Markdown file, just so they don't show up when I knit this together and export it to put it on RPubs. So I'm over my chunk. Okay, I did actually remember to install APE, so it's ready to go. And now I'm ready to take the files from Canvas that are very workshop-y files, a super small FASTA file. This was actually one of the FASTA files that was uploaded as part of a previous incarnation of the Genes and Genomes class. So this file came from our sequencer at Endicott. A FASTQ file, I looked through the entire American Gut Project for some data that wasn't high quality. So this one was perfect as a workshop model. It only has 3 clusters that pass filter. So there's only 3 individual reads in this FASTQ file. A typical MySeq run will give you 15 million. So this is a weird file, but it gives us great practice, and we don't have to worry about taking up any space on our computer. And then the last one is a variant call file. This actually also came from an example that I made on Galaxy. So this is a workshop type example that I have in my Galaxy history, where the end result is only 2 variants. So again, a normal variant call file would be much longer. You'd have a lot more information, but all of these small files were things that I chose for how small they were and how easy they'd be to work with to see the structure without taking up too much space on your computer. So the first thing we're going to try to import is a FASTA file. It's pretty easy to do. There is a read.fasta function that is built into APE, and it works really well. So your final knit HTML file will be longer than this because you'll need to put in comments and explain what you're seeing at every step. But otherwise, the commands can be the same. So when you read the file and you run the head command and you run the structure command, you're going to see this really nice report that shows up from the head command. When you have read in the file with read.fasta, the kind of data that it gets stored at is a list. So you can see the list here. You can click on the blue dot if you're in RStudio to get an idea of how the data is being stored. But you can also, look at this report when you click the head command and see, okay, well, the sequence is 1,560 bases long. This is the base composition, and this is what the label is. So this is the whole FASTA header up to a certain number of characters, and then it just adds the ellipsis, ... When you run the struct command, it tells you that it's a list of 1, and it prints out the entire header and tells you how many nucleotides are in the count, and then does a preview of the binary data that's being stored for each base. Really simple commands, but they give you a lot of information about what you've just read in. For FASTQ, it's going to be pretty similar. You're going to need to comment this and explain what's happening at each step, but the built-in read FASTQ file works very well and should also generate a binary list. Those two functions share a lot of the same commands if you want to look them up on GitHub. And then the last one is the variant call file. And this one is going to take some troubleshooting. So I showed you the first couple of steps as to how I would approach reading in a variant call file. Read.table works really well for ignoring all of the commented characters in the variant call file. So if we look at this, you'll see most of the first lines have 2 pound signs, 2 hashtags in a row. And so all of those 13 lines will be ignored, and read.table will only try to read in lines 14 and 15. Read.csv is really useful because there's information in those commented lines that you might want, specifically what to call each of the column names. So you have two choices here. You could either use the name commands and manually type out what you want the columns to be, or you could do something like line 33, and you could read in all of the file, keeping those lines that are commented out, and then do something with the 13th line so that you can pull the variant names, the variant call file column names out of that line 13. So I'm just going to run this command just so you can see what I'm talking about. You can see that when you run the structure command, you get like a little preview of what's in each of the columns. You can see what data type is in each column. You may need to fix that for some of these. And then you can see the column names right now are just V1 through V10. But if we look at my lines, we can see line 12 has what they should be called. So chromosome is the first column, position is the second column. And it's up to you whether or not you want to type your own names or try to use my lines in read.csv to get that automatically filled in. If you have any questions, please reach out via email. I'm looking forward to seeing how you guys are able to describe this process. I think of this as being very similar to the undergraduate assignment in undergrad bioinformatics of like what the FQ, trying to figure out what all the different file types are. Here, you're just comparing the information. So your comments should be describing what's the file type when it's stored on your computer, and then also what's the data structure that it gets stored in R. So good luck with this assignment, and let me know if you have any questions.